Class, today we were supposed to take an adventure inside the human body, but unfortunately the bus broke down today, so we're going to learn via a presentation.
Mr. Augustine, what are we learning about today?
Thanks for asking, Ali. Today we will be learning about how a glucose molecule travels through the cell.
What? Why is glucose traveling inside of me?
Johnny, glucose is actually a sugar, not anything to be scared of. It's in your food, so it comes into your body through your mouth, to the esophagus, the stomach, small intestine, blood, body tissues, the cell, and finally inside the mitochondria to produce energy.
So when the glucose gets to the mitochondria, what happens?
Great question, Johnny. During this process, two ATP molecules are generated through substrate-level phosphorylation. When phosphoenolpyruvate donates a phosphate group to ADP, forming ATP. The formation of 2-NADH occurs when NAD+ accepts electrons from G3P in a redox reaction facilitated by the enzyme G3P dehydrogenase. Two molecules of a pyruvate are formed through the splitting of a 6-carbon glucose molecule during glycolysis in the cytoplasm of the cell. Can anybody explain acetyl-CoA?
Oh, definitely not. Who knows all this stupid weird stuff anyway?
It's not weird, Johnny, and yes, I can explain it.
Yeah, it's not weird or dumb at all. And of course, go ahead, Ali.
Alright, bet. During the transition reaction preceding the Krebs cycle, one molecule of pyruvate undergoes oxidative decarboxylation, producing two molecules of acetyl-CoA to enter the Krebs cycle. This process results in the release of two molecules of CO2 and the formation of two molecules of NADH through the reduction of NAD+. Mr. Augustine, there's more than just that, right?
Of course there is. Next up is the Krebs cycle. In this cycle, a 2-carbon compound combines with a 4-carbon compound to form a 6-carbon compound. Through a series of reactions, the 6-carbon compound is gradually oxidized and broken down, regenerating the initial 4-carbon compound, ready to combine with another molecule of acetyl-CoA. This cycle occurs twice per glucose molecule to process the 2 pyruvate molecules derived from glycolysis. In total, the Krebs cycle generates 2 ATP, 6 NADH, and 2 FADH2 for each glucose molecule. This process takes place in the mitochondrial matrix.
Oh wow, this is kind of actually interesting.
Okay, Johnny, this time I want you to do something. Can you at least do a basic Google search and find out what oxidative phosphorylation is?
Sorry, Mr. Augustine, but I can't be doing all that. My phone isn't connected to the Wi-Fi.
You for real?
I'll do with that. Here's what it says. The energy from glucose is stored in the form of high-energy electrons carried by NADH and FADH2 generated in glycolysis, the transition reaction, and the Krebs cycle. In the electron transport chain, redox reactions involve the transfer of electrons from NADH and FADH through a series of protein complexes, gradually releasing energy at each step. Oxygen acts as the final electron acceptor in the ETC, driving the redox reactions by accepting electrons and protons, ultimately forming water. These redox reactions power the pumping of protons across the inner mitochondrial membrane, establishing a proton gradient. The proton motive force drives ATP synthesis by allowing protons to flow back through ATP synthase, a process that harnesses the energy from the proton gradient to convert ADP to ATP. Generally, oxidative phosphorylation produces approximately 28 to 34 ATP molecules per glucose molecule.
Oh wow, this is really cool, Mr. Augustine. So what does all this ATP do?
I love that you started to gain interest, Johnny. This ATP can be used for many bodily functions. This can include intracellular signaling, DNA and RNA synthesis, and many more. The energy derived from ATP hydrolysis provides the necessary fuel for the enzymatic processes involved in DNA and RNA synthesis and intracellular signaling.
Wow, glucose is extremely important for the body.